Hey everybody, in this video I want to talk some about the cardiovascular system, particularly the big picture overview of the plan of the circulatory system, as well as the different structures of the kinds of vessels that we have in the blood vascular system. And then at the very end we'll talk a little bit about distribution of the blood throughout the body, because it'll be different when we're at rest as opposed to when we're more active. Now, this cartoon illustrates the basic system of the cardiovascular and lymph systems. Cardiovascular really means blood vascular system. So when we say vascular, we're referring to vessels. Now, this in this case, what we see is the heart in the center here as a pump that pushes out blood first through the big elastic arteries, and then those big arteries will split into smaller distributing or muscular arteries, and eventually to get out to capillaries in many cases. But sometimes they'll also just go directly back into small veins again, and eventually into large veins back into the back to the heart. You'll see kind of thrown in green here this lymph vascular system that's thrown in as well. The lymph vascular system we're going to talk about more in the next chapter, but it's sort of a fluid balance system that also has some functions related to the immune system. So we'll talk about its importance a little bit later on. But first we're going to focus on the structures of the blood vascular system. So when we consider the blood vascular system, there are some things we know about them. We know that all the arteries go away from the heart and that they branch. We also know that all the veins kind of merge, they come together, and they also are all headed toward the heart. One thing that all of the big vessels, arteries and veins, have in common is a series of 3 tunics. These are like the strata of the skin or the zonas of the adrenal cortex. These are the different layers that are characteristics of each. So if we go outward let's start with arteries. If we go outward for the lumen, we see that first lining the space where the blood is a thin layer of tissue that we call endothelium, this inner part of the tunica intima. Outside of that, there's this connective tissue layer called the subendothelial layer, and then lastly an internal elastic membrane. That internal membrane is there to help arteries resist the big changes in pressure that happen between adjacent ventricular systoles. External to that is a layer of smooth muscle that's called the tunica media. The tunica media is what's going to be responsible for maintaining the diameter of the artery. And then external to that is another elastic membrane, which is, again, important to help to give you kind of passive support against blood pressure. In the tunica externa, it's mostly connective tissue like collagen fibers, but very large blood vessels will actually have their own arteries that go to them as well. They're thick enough that they can't get what they need just through diffusion. That's what's labeled here as the vasa vasorum. Now, blood, when it goes out through arteries, starts off in large, what we call conducting arteries or elastic arteries, and then goes out to smaller muscular arteries, or what we call distributing arteries. And those are what's going to be bringing blood to making decisions about what parts of the what capillary beds are going to be getting blood and which ones are going to be kind of closed off. More about capillaries in a bit. The very smallest arteries we call arterioles, and they're going to go out to into capillary networks. I suppose we'll talk about capillaries now. Capillaries are thin. They are thin-walled. They don't have the same 3-layered setup as what arteries and veins have. Instead, they just have a single layer of endothelial cells that will differ with the different types of capillaries, but those thin layer of endothelial cells are going to make it easier to allow diffusion from the blood into the interstitial spaces that are around the cells of the body. Then surrounding the capillary, there's the endothelial cells. There's a thin basement membrane, which you might recall from our early discussion about tissues. A basement membrane is like a protein sheet foundation that those endothelial cells will have to give them a little bit of support and connectedness to one another. The capillary is shown at hugely exaggerated scale. You might recall from looking at this under the microscope in lab that capillaries are small enough that red blood cells have to go through them single file. Now, capillaries, they tend to occur in beds, as sort of shown diagrammatically in this cartoon. They're going to merge together into small vessels that are called venules, which are then going to join up with larger veins. Now, veins are similar to arteries in that they have 3 layers of tissues that support them, but they're a little bit more stripped down. Notice that they have an endothelium. There's this subendothelial layer, sort of like a basement membrane, but notice that there are no elastic membranes, neither internal nor external elastic membranes in veins. And also, the muscle layer is a lot thinner than what we see in arteries. And again, that's for reasons. I mean, muscle is expensive, so why burn metabolic energy if all of the blood vessels of all the veins are sending blood just back to the heart again? So they're a little bit more stripped down because they don't need to be like making decisions about shunting blood from place to place like small arteries and arterioles do. One addition that we see in veins is the presence of valves. Now, those valves are one-way valves, just like we talked about before with the heart. And what they do is they keep blood kind of limping forward toward the heart, even though it's under low pressure. Without these valves, then blood would really pool in your lower extremities, just because gravity would pull them that direction. I guess the last thing to throw in here is, I said before that arteries and veins differ in the thickness of their smooth muscle. That smooth muscle allows them to kind of, when they tense up, that's going to shrink up the diameter of the blood vessel, and we call that vasoconstriction. So vasoconstriction is narrowing the lumen with muscular contraction, and vasodilation is the opposite, where the lumen is expanded. The difference in the shapes of artery and veins, I know we've already seen in lab, but just to kind of remind you, arteries tend to appear roundish in cross-section, whereas veins tend to be a lot flatter. This table here, I'm not going to walk through exactly, but it's a good summary of what we've seen for the 2 types of big arteries, elastic and muscular, as well as what we see with veins. And and they also make a point to say that these are not shown all at the same size, but the relative proportions are reasonably accurate. Okay, now I want to talk a little bit about the different kinds of capillaries. These are important because though capillaries we know are the most numerous kinds of vessels, they are different from one another depending on the places in the body where they're found. The words that I'm going to kind of go through are found in your textbook, of course, but I'm going to zoom in just on the figures here so that we can distinguish some of these differences of capillaries. First, I would say that the functions of all capillaries is where exchanges happen between what's in the plasma or gases that are carried by red cells. It's the only place where exchanges happen between the blood and the interstitial spaces where the fluid is outside of the tissues. We will see that there's sort of a water cycle in your body, and the water is going to give different names depending on where we're looking. For example, the water that's inside the blood, we call the plasma. The water that leaves the plasma and crosses the blood-brain barrier, we call that stuff cerebrospinal fluid. The water that leaves the blood and goes into tissue spaces, instead of calling it plasma, we call it interstitial fluid. That fluid, once it goes into a cell, we then call cytosol. So, and we're also going to see later that interstitial fluid that doesn't get absorbed back into the blood vascular system will get absorbed by the lymph vascular system, and at that point we call that liquid lymph. Okay, so there are a whole mess of different names that we're going to have for fluids that are distributing solutes to the cells. Okay, anyway, so now about the different types of capillaries. The most numerous type of capillaries are called continuous capillaries. They have all of these capillary types are joined up by tight junctions that are labeled here. Tight junctions are permeability barriers. They prevent liquid from leaving very easily, although in some places there'll be clefts, larger spaces between the cells as well. But what actually leaves the plasma is regulated by these cells that sit on top of the capillary that are called pericytes. And those pericytes are responsible for, like I said, for regulating what's going to go in or out. The continuous capillaries they're most abundant in the skin, in lungs, in muscles, in the central nervous system. Although the ones in the brain are unusual, because they help to form that blood-brain barrier. They are totally enclosed with tight junctions, and they don't have any intracellular clefts when we're looking up in the CNS. Okay, so this would be like the most typical one. So only the very smallest solutes are going to move, largely by this process called pinocytosis. We talked before about exocytosis with neurotransmitters leaving the end bulbs of axons. Pinocytosis is a similar process. It's another membrane regulated process, but this is what they call cell drinking, you might remember from Bio 111, I guess. And this is for allowing basically little small sips of the plasma to get drawn through the blood cell, or the endothelial cell, to be able to kind of go through through the pericytes in order to get to the interstitial fluid. But compare the structure of continuous capillaries with what fenestrated capillaries look like. So they have all these holes in them. So they will still have pinocytotic vesicles that are present to allow a lot of exchange. But one of the things that's different about fenestrated capillaries is this is found in places where you have filtration that happens. Now, filtration happens primarily in the kidneys. So these fenestrations are small enough so that solutes can get through really easily, but nothing as large as a whole cell. Nothing's large as a whole eukaryotic cell, and also too small for bacteria as well. So they're allowing you to kind of squeeze through fluid, squeeze through fluid like through a sieve in order to sample from it. We also find it in the small intestine where there's a lot of absorption that happens. Again, this helps to facilitate that absorption. They're also found in lots of places where hormones are produced as well. So where you have cells that are actively producing substances that are going into the plasma, they're going to get picked up through these fenestrated capillaries as well. I suppose I should throw in just the idea that fenestra is a Latin word that means window. Okay, so these are little windows inside the capillary beds that are going to allow easier diffusion. The last kind, you can sort of see the end point of this. With sinusoidal capillaries, these are the most permeable of these vessels. It says here that occur in special locations, largely places where whole cells need to get into or out of the bloodstream. For example, in bone marrow, this is where red cells and white cells and the other formed elements are going to platelets, I guess, are going to be able to leave the marrow and get into the circulatory system. So they got to be big enough to pass through these larger voids. The Other place to find them is in the spleen. We'll see the spleen is sort of a it's kind of a dumping ground, kind of a reservoir for red blood cells, and also lots of white blood cells reside there too. So they need to be able to transit in and out of the blood. The liver is important there because the resident macrophages that live in the liver, what they will do is they will send their little tendrils, send their little dendritic ends into whatever their targets are to be able to kind of detoxify the blood. So we find them in particular places like that. The blood flow tends to be pretty slow through them, which is another thing that allows sinusoidal capillaries to be able to let big things in and out. Okay, a little bit about microcirculation. The blood, once it reaches capillaries, there are a few different things that can happen. Now, we've talked before about in class how like the heart gets what it needs and the brain gets what it needs, but everybody else like takes turns. So not all of your capillaries will be open at all times. This is regulated by these little sphincters that are on the arteriolar side of the capillary bed. So in the cartoon here, you notice in the upper panel, this is with the sphincters open. So when that happens, the blood that goes through the arteriole is going to spread out into the tissue bed through this capillary plexus, and then they'll all kind of merge together onto the venule. On the other hand, if this is a tissue bed that either has been like deprioritized in preference to other structures, or if this is just tissues that aren't particularly oxygen starved or food starved, then those sphincters can get closed off. In that case, blood will go through this thoroughfare channel. This mid-arteriole will allow a little bit of diffusion, but not a ton. Even though it looks like it's just going straight red to blue in the cartoon, you'll get a minor amount of diffusion, but not too much into the area. So this, the relative amount of blood that's in a tissue is something that we call perfusion. High perfusion happens like in the upper panel here, where we see lots of blood getting sent to this tissue bed through the capillaries, but we have low perfusion in the lower panel, where a relatively small amount of blood is passing through. Just a little note on veins here while we're kind of going through. I've been through a capillary bed. Recall that those valves, they're important for keeping blood moving in the same direction. But when you consider all those veins that are like in your legs and in your abdomen, blood's going uphill most of the day, right? So in order to allow that, veins kind of take advantage, because like whenever veins get squeezed from the outside, they have it allows the blood to move forward kind of passively. We'll see in a moment that the blood pressure is actually really, really low by the time it gets into like the vena cava. But the reason why it's able to keep moving forward is because muscles that are around, like regular skeletal muscles that are around the veins, will squeeze on them from the outside. And when that happens, you figure the blood gets squished in both directions. The blood in this picture that gets squished down would close the valve. The blood that gets squished on the other side is going to open up the valve on the other side and eventually kind of leapfrogging its way up to the heart. In addition to the muscular pump, which happens, due to skeletal muscles, even just ventilation, breathing in and out, the pressure changes that happen in the chest, that is also enough. We call that the pulmonary pump as well. Those are both enough to be able to move the blood forward to be able to get back to the heart. Now, veins have relatively large diameters, pretty large lumens and thin walls. So they're what we call capacitance vessels, which means they allow place for blood to get stored. So just skipping over a slide here. In this case, at rest, I think this is at rest, most of your blood is in these large systemic veins and then the smaller venules that lead to them. When you are actively exercising, like when I made you run the stairs, in that case, the veins will tend to have, they'll have a little bit less, and there'll be more going to the heart, more going to capillaries, more in the systemic arteries and arterioles. So I think so this distribution will change, but largely the veins are there to be able to kind of hang on to the blood that's kind of limping its way back. We'll see, one of the things you should understand is that not all of the blood goes through the heart like at the same rate. Some blood will be making relatively quick circuits through the heart, just depending on the route that it takes, whereas some parts of the blood are going to go on a much longer time frame to be able to get through the heart. And just to go back here a little bit, I sort of like this summary of different kinds of blood vessels, although it isn't a picture that's in your textbook. Just to kind of work through these, compare the difference between the upper left and the lower left panels, parts A and B here. Notice that the upper one's showing you the individual vessel diameter. So we see that the aorta, that single aorta, has individually is a very large pipe, and goes out to these smaller muscular arteries, which are individually smaller but like way more numerous, or just at least more numerous. The arterioles are smaller still, and then eventually the capillaries are the very smallest ones. And then as you see again, as the blood is kind of re-merging back to into larger and larger vessels, eventually the two vena cavae together are larger than the aorta is. On the other hand, when you consider the total cross-sectional area of all of the vessels of a given type, you see that the aorta is a relatively small part of the total cross-sectional area, whereas as are the vena cavae. But notice here that it's the capillaries and the vessels that are immediately going to the capillaries and away from the capillaries. Those are the ones that have the greatest total cross-sectional area. Compare that then with the average blood pressure. Notice how the blood pressure starts off really high, and we'll see later it's more variable too, where it's close to the heart in the artery side, but then it starts to slow down. Then it's the pressure goes down, down, down as you are eventually limping your way back to the heart on the other side. So when we measured blood pressure in lab, we were measuring like the pressure in these pulsatile elastic arteries, like the brachial artery, not that really low blood pressure at the vena cava. Another thing that's related to this and important for exchange in capillaries is also the velocity. So blood going out through the aorta is moving really fast, but as those muscular arteries split apart and split, what happens is the velocity will slow down until it's very slow in capillaries. But then as those vessels kind of merge together, and as there are fewer of them, it'll start to speed up again overall by the time it gets out to the vena cava. Particularly the two bottom panels here, the total cross-sectional area and velocity of blood flow, with those, what I take away from those is thinking about the circulatory system like streets, like roadways. And when you think of how many lanes there are on like I-93 or I-90 into Boston, there are relatively few. It's relatively few lanes there if you compare them to like the width of all of the lanes of all those streets in the city of Boston. So the total cross-sectional area is like the number of lanes in a given area. So your interstates would be on either end, the elastic arteries and vena cava end. But the city streets, like all the little winding streets on Beacon Hill or South Boston, or the North End, those are what the capillaries are like. And so think about it. I mean, you got like the world's worst boyfriend if he's making you get off on the highway. But usually, when you want to drop somebody off at their house, of course you're going to slow down and probably even stop the car. And so that's one thing that having a large cross-sectional area does, is the blood kind of like the pressure is lower down, and also the velocity is slower, so that the exchanges can happen in those capillaries. Whereas in the bigger vessels, it's just about bulk movement of the blood. Anyway, that's the main stuff I wanted to get to for this part. Tune in next time for talking about regulating blood pressure.